the way that we've set this meeting up is we'll try to get through most of the introduction and get through most of the data in the first hour and leave a good hour for discussion, because I know at the last meeting we ended up having to rush through it at the end, and you know, I think this time we should have plenty of time for discussion. So I think as we go through the slides, let's make sure that, you know, if we want to stop and ask questions, let's do that. All right. So without further ado, I think, you know, we'll get started, and I think the rest of our team will join us when they can. So let me just get this. Okay, so the agenda is shown on this screen. So we're going to have just a very brief introduction. I'll go through the kind of action items from the previous meeting that I always do, and we'll do the SGC Oxford update, the Charles River update, the OICR update, and then I'll come in and talk about some of the things that we're doing to profile the lead molecule, M4K 2009 at this point. And then, as I said, we've left plenty of time for discussion. So let's make this a discussion-based meeting if possible. All right. So the action items from the previous meeting that I captured are shown here. Alexi was interested in some of the newer molecules that we had highlighted at the previous meeting, and to run in his RIPK2 assays and for MTT testing. And David Schmiel and I organized that and got the compounds sent out. Jerome, we wanted to provide him with some of the newer compounds as well that we've identified, as well as a key reference compound that he can include in his assays that is structurally similar but completely inactive against ALK2. So it's always a good control to include in especially the phosphosmad type assays. We wanted to continue to prioritize compounds for X-ray, and I think, you know, we're doing that. We're you know, Oxford is trying a bunch of different conditions, and, you know, perhaps we'll hear a little bit more today from Alex. I know that there has been some issues with protein and various issues, so we'll see what's happening there. We had discussed doing a more thorough IP search on M4K 2009 specifically, where we've introduced that C4 methyl in the pyridine. And this was something that Max, Owen, and myself were looking into. We had some discussions around it, and, you know, I'll touch very briefly on at least some of the things that we've done in the past month on an upcoming slide. Jerome was going to check with Stanford about accessing additional cell lines. Unfortunately, Jerome can't be with us today, so he, you know, we won't get an update from Jerome. But you know, I think next month we'll have a very interesting meeting with a lot of updates from a lot of key people. We wanted to finalize the MTA with the DIPG consortium with Javad, and I just saw an email yesterday that that has been done. And I think we're ready to go. We're ready to send a compound to Javad. So I'm just waiting to hear from him about how many compounds he can handle, and you know, how much compound he needs for the various assays that he's willing to run for us. So I think that's great. MDCK, MDR1 assay is, again, I think still close.
It is. So we got new cell line from Sigma. The older cell line, we had all the assays running. Mm-hmm. But the data were not really corresponding to the literature values. These mm-hmm. are control compounds. So we have new cell line from Sigma. We're in touch with them, so we are going to run them again. Most likely in the middle of September, because it takes about we are splitting the cell. Now we are going to grow them. It takes about 4 days to grow the plates. So most likely next time we can report what happened with this cell line. So if it is working, then we'll be ready to run.
Okay. Great. Yeah. No, I think that's great. You know.
Thank you very much. We'll get back to you by tomorrow about the amount of compounds that we need, and looking forward to working with you.
Okay, great, Javad. Nice to hear that you're attending, and yeah, we're certainly looking forward to working with you and your team as well. So, I think certainly M4K 2009 is one that we have plenty of material to be able to send to you. And then the other ones, we can discuss further as we move forward. All right. And then, I know that, this has been on this number 8 has been on our list for quite some time now. Genna is working hard to be able to finalize this for the ALK2 project and a couple of other projects that we have running internally. So hopefully very soon we'll be able to have this database up and running and be able to, share all of the data that we're generating for these compounds in one concise place. So with that, I'll move forward. So when we met last time, there was some discussion about evaluating in a more extensive fashion the IP, the IP and the freedom to operate around M4K 2009 specifically, where we've introduced this C4-methyl. And there was a suggestion made that we register the compound in the PubChem structure database, which is a publicly available database that anyone essentially can register new compounds in. And so we've done that. The link to the compound is here, or to the database is here. I won't click on it because basically you'll just see this kind of screenshot. It highlights that it's a compound from M4K Pharma, that it's designed for DIPG and targeting ALK2 or ACVR1. So again, this is kind of a mechanism to ensure our compounds are found in any patent examiner's search. And also, in order to kind of protect our freedom to operate around this scaffold, we're considering a very early publication. And this is something that I think, we can discuss at the end of the meeting, or, since I think Owen isn't at this meeting because he's on vacation, he might be dialing in, but he's not able to be here in person. And I'm not sure that Aled is able to make it either, because I know that he's on vacation. So I think that, this is a discussion that perhaps we could save for September's monthly meeting. But I did want to just highlight that we have started releasing publicly some of our structures to be able to protect them as well as we can without, of course, issuing a patent. So with that, that's really the end of my intro for this week. And I think we can jump right into Alex and Jung-Fu's presentation. All right. Which I can pull up. There here. All right.
Thanks, Jeff.
Yeah.
Alex, I'll just say a few updates from Oxford, and then I'll hand over to Jung-Fu, who can just kind of run through this one slide, some of the cell data.
Yeah.
So some of the sort of actions from Oxford, we've also just recently sent some compounds to Alexi for the RIPK2 collaboration and project. We've also just sent some kinase plasmids to Kate at Reaction Biology, which is also part of a collaboration. We've been doing a lot of discussion with Promega, SGC, UNC, and David Drury in particular, and colleagues about building a tracer for the ALK5 nanobret. And so with yourself, Jeff, as well, we've got some discussions with David there about potential design ideas. And if we can get the ALK5 nanobret, then that would be a fantastic matching assay for the ALK2 work that's shown on this slide. So hopefully that can come on shortly. And also very grateful to everyone at CRL. We've got access now to the database. So hopefully if that makes life easier in terms of sharing data and documents, we can do that. And then on the crystallography side, yeah, we've had our baculoviral viruses for expression. For some reason, over time they must degrade, and we've lost 2 of our key viruses. But the team has been going to other constructs that we've had and trying crystallizing those. We will regenerate those viruses, and I think that's probably nearly done or being done.
Do you want some updates?
Yeah.
We're going to do a titration of the construct 96, a titration of the virus for Friday, and construct 76.
So that's nearly, well, nearly there.
Isn't really
but
working.
We will in any case be sending up plates with whatever we have, and in the future back to the constructs that we know have worked well. And the synchrotron, where we would collect our diffraction data, is closed for summer maintenance and upgrades. So even if we had crystals this month, we wouldn't be able to collect any data. So unfortunately, we're going to have to pass on that for this month and hopefully come back to it for a future meeting. So I'm going to hand over to Zhongfu. He'll just give a brief update on some of the cellular nanobreath.
Great, thanks, Alex.
Hi, Jeff.
Hi, Zhongfu. I was sorry to hear about your knee there. I hope you're healing up. And,
thank you.
Wish you all the best.
I have only myself to blame, no. So the previous batch of the compound, there are around a dozen of them in total from OIPI and Charles River's lab, and among them 2 are quite interesting. Even with the replacement of the nitrogen with the fluoro and the hydroxy group, I think I've compiled multiple experiments for the reference compound M4K 1062. It doesn't really compromise the potency towards ALK2, so I'm quite excited to find out what is the off-target activity towards ALK5 would be. Although in this batch, none of them are as potent as the previous batch, where we've gotten one with the Nitrile substitution that had 20 nanomolar IC50 to ALK2, but definitely I'll need to perform more cellular assay to determine the ALK5 activity to see whether the increase in potency towards ALK2 it compromises the specificity. Yeah, but I'll do that as soon as possible.
When are you doing the ALK5 assay?
Next week.
Okay.
Next week? Okay, great.
Do we have some compounds from the previous month's meeting to also do by ALK5?
Yeah.
Okay.
I'll be checking them at the same time.
Okay. So we've got a summer student here as well for the next 2 months. That's Karina, who's just joined. And hopefully she'll have be an extra pair of hands that we can also expand Zhongfu's capabilities. So
considering I'm somewhat handicapped.
Well, welcome, Karina.
We were thinking, maybe the scope to also look at nanobreath of ALK3 or ALK6, some of the other BMP receptors, or ALK1. So we'll see what we can do there. If there are any other kinases, whether it's RIPK2 or Diable, we also have nanobreaths for those. So if there were a priority set of half a dozen compounds even, we could do some wider nanobreath screening.
Yeah, I guess, Alex, I think along those lines, we have tested M4K 2009 against the full kinase panel. So, I'll show those results again in my slides. And maybe if you have any of the nanobreath tracers and assays up or ready to go for some of the kinases that we hit in that assay, it might be interesting to get cell-based data.
Yeah, we have a DDR1 nanobreath assay as well, and we have seen differences in some assays with cell data and the kinase assays. So
right, that happens a lot. So yeah, I think I mean the DDR1 was one of the things I noticed that you're on a publication with about a DDR1 inhibitor, so. I kind of felt that you might have some of those assays available, so it was one of the comments I was going to make when we go through it. So
that one's a surprise because, you know, we've made a bunch of these. We've done previous kinase selectivity profiles, and we know some kinases crop up again and again, like RIPK2. But I have a feeling this is the first time we've hit DDR1. So I have a feeling it won't be a common off-target. So I'll be interested in this SAR series, you know, if we do some future kinase profiling, if it retains. Probably, I wouldn't be surprised if we can get rid of that one.
Yeah, and I don't think it's a big liability anyway, right?
No.
To be hitting that very hard. So anyway, I think, you know, when I show the data so that everyone's on the same page, maybe we can circle back to this discussion.
Okay.
Yeah. All right. Okay, great. Thanks for the update, guys. Let me just set this up. Sue, are you ready to go?
Yep.
Let me see. This one.
That's the one.
All right. That's the one. Done.
Okay. So just in terms of a general update, we're continuing with our chemistry. We've got a few extra people helping out at the moment. We've had some new recruits, so we're hoping to make a little bit more progress in the next month or so. Obviously, we'd still like to get the crystallization data on the M4K 3007, the blueprint compound, because I think we're sort of interested in getting some further, see if we can develop a second series perhaps based around something similar to that structure. But we've done some redocking because we docked originally, and I'll show that on a later slide, but we'd docked one of the compounds from the blueprint patent, and it didn't happen to be M4K 3007. So we went back and redocked that and did see some slight movements from the docking, and it's given us a little bit of a few inspirations for some new ideas, which I've got on a later slide. So obviously and the other thing we've done, we've completed all the plasma protein and brain tissue binding, and I guess you'll cover that in your slides later, Jeff.
Yeah, I sure will. Yeah, thanks.
Talked a little bit about it last month, but we didn't have we didn't have brain tissue binding. So on to the next slide. So this is just a reminder of the blueprint compounds. So 3007 is the most potent of the compounds that we selected. 3008 doesn't have either the pyridine nitrogen or the ethoxy group on the piperidine, and is about tenfold less active. And then replacing the cyclopropylamide by the urea has resulted in another threefold drop in potency. This is quite surprising because when we do the dockings, this cyclopropyl ring sits in a very hydrophilic region, and we thought that, well, maybe the urea might be preferential in that position. So those are the results from the compounds. They're all pretty selective over ALK4 and ALK5. So it's interesting to see how selective they are. But obviously, M4K 3007 remains to be the most interesting of these. So we go on to the next slide, please. So the original compound that we docked actually was M4K 3008, which, as you saw previously, was about tenfold less potent than 3007. So if you look on the right-hand side, the compound in the pink, which shows the interaction at the hinge, the carbonyl, the cyclopropyl carbonyl interacts with the lysine, and then the piperazine phenyl group goes out towards solvent. And when you put it looks like when you put the pyridine nitrogen in place of the phenyl, that you get this movement, so that pyridine nitrogen now interacts with the tyrosine. Hopefully, well, this is purely from the docking, and obviously we would ultimately need to have this confirmed from the crystal structure, but it is quite interesting that you see that movement.
Yeah, that's quite a striking movement. that's quite a big movement. Just yeah.
It is. So the methoxy group doesn't appear to actually do anything, interestingly. So from the docking, it looks like the pyridine is the key feature, and obviously that would suggest that, as far as I could tell from the compounds that have been made, I don't think we've made the compound at the bottom. It should be a methyl group in the middle there, but with the pyridine nitrogen in that position, as far as I can tell. No,
we've made the one with the nitrogen at this position.
Yeah, exactly.
But
which
not
is probably less likely to be able to give that interaction with that tyrosine, looking at where it's positioned. So it's just a suggestion that maybe that could be added to the target list.
Yeah, I think that's a good suggestion. I mean, we should cover all of our bases anyway with the pyridine permutation.
We're happy to take that on if you would like, but it sort of fits more. I mean, it's up to you. If you would like us to make it, we can add that to our target list.
Well, I think, from what I've seen, your target list is pretty full. We have the starting materials already available here, exactly. So I think it makes sense. That's what I thought.
Yeah, absolutely. No problem.
But we'll definitely add it. Yeah.
Okay.
It's a good suggestion.
I mean, one of the things we had about 30 seconds to look at the docking model this morning, and one of the things I noticed, the 3-membered ring does interact with the phenylalanine that folds in from the glycine-rich loop.
Right. Okay.
I don't know whether there's any hydrophobic interaction there. It's probably minor, but we suspect there will be a difference there between ALK2 and ALK5. The phenylalanine, I think, is a tyrosine. No, maybe, I'm not sure if it's a switched tyrosine, but it has a more tendency in ALK5 to swing outwards. So that hydrophobic interaction, even if it's minor, might be more ALK2 selective, but we'd need to have another look at it to see if it was very significant.
It wasn't that clear to us, because it looked, so we had to do the docking in the absence of water. As soon as you do it with the waters present, it's really hard to get it to dock. So I don't know whether they're waters that are displaceable or what, but that is a very hydrophilic region, which is why we were quite surprised that cyclopropyl sat in that region.
Yeah.
So it's hard to explain, but it looks like it's interacting with the phenylalanine. Did you say, Alex?
Yeah, I forget the number off the top of my head.
I'll have another look at it and see.
I think from all the crystallography we've done, the yellow molecule on the top right looks far more believable.
Yeah.
Okay.
Yeah. Okay.
Okay.
So moving on. So this benzimidazole we looked at last time, and it's got moderate potency, but when you compare with, say, the indazole, M4K 1126 there, you can see that it's actually about between 3 and 4-fold more potent. So putting in the additional methoxy may well help with that activity. And there's a few other suggestions around there that we've got. Obviously, we haven't made the compound with the 4-methyl on the pyridine ring. That's in progress. Hopefully, that will drop out fairly quickly. And we've got compounds with the methoxy group on there, and moving the benzimidazole around one position and putting in either a nitrogen or a fluorine on the linking ring. So a number of ideas that we've been working on. They're all well along the way. The guys have been making significant progress on these. It's just they've not quite got to the end. Some of them actually putting some of these benzimidazoles with methoxy groups in. It's a few steps to get to them. So hopefully we'll complete the synthesis of some of those in the next month. Obviously, we are in the holiday season.
Well, of course. Yeah, us too. Yeah.
Yeah. Although I don't go on holiday till the end of September, so. Okay, so we'll continue with those. And I also think it's worth looking at things like the nitrile, because I noticed that nitrile. So when you make the compound with the nitrile, I'm just interested, is that made with the nitrile in situ, or do you put that in at a later stage?
That's actually, it's in the starting material. So we buy that.
Yeah, I saw that was commercially available, and I thought that was probably worth having a look at. So we'll try and make a few of these compounds with both methyl and nitrile in there. Thank you.
Yep.
Great.
Okay, next. So these are sort of to follow up on 2009 and 2106. We've talked about this before, replacing the methyl or nitrile by a carbonyl. This compound shown in the yellow box at the top. Synthesis of that is, those are in progress. We've got things like the intermediate benzyl compounds where we've protected the pyrimidone in order to facilitate the Suzuki chemistry. So we've got those intermediates. We've just got to deprotect and then put the relevant group on the other side, if that makes sense. So making the two compounds. At the bottom, we've started making these pyridones. So these come through from corresponding fluoropyridines. So the two fluoropyridines, the hydrolysis of those. Again, we're nearing completion on some of those targets. So there's a lot of things that are.
Sue, will you submit can you submit the fluoropyridines as well?
We can if we've got in. I'll check whether we've got enough of them. Yeah.
Okay.
Okay. Yeah. I'll just check whether we've got enough, and if we have, we'll submit them. Yep.
Okay, that'd be good.
Okay. And on the next slide. So these are some things I talked about last month, where we're sort of accessing the Nicene Pocket from the 4 position. So this is starting to move away from the main series with the idea of potentially generating a second series. So either having a linker such as an oxygen linker or a carbonyl linker. So the pictures on the right, the docking pictures on the right-hand side show the carbonyl, which has an interaction with the asparagine, I think it is. And again, synthesis of these is in progress. Again, these are being prepared by some of our new starters that have just joined us very recently, so which is great news. So they're making reasonable progress. Yeah. So, and finally, these are some more new ideas we've had, and this is trying to use looking at the blueprint compound and comparing it with our series and doing some overlays, and actually looking at replacing the trimethoxy phenol by something like a Perazim with a cyclopropyl amide on the end. If you look at the bottom left-hand picture, this is a docking. It sits in a very nice similar position. Although it doesn't put the cyclopropyl in the docking in exactly the same place, you can envisage that you could get some rotation. Yeah, it looks like that they ought to sit in similar places. So I think that's well worth looking at. We could make a series of amides to explore that pocket.
Yeah,
I put the cyclopropyl amide up, but I think you might make a small set of amides, you know, assuming we can get chemistry to work with a Bock in place on the other end.
Yeah, sure.
Then looking at the benzimidazole type compounds, yeah, that one. So here you don't have the carbonyl, but this places that cyclopropyl in exactly the same place as the cyclopropyl in the blueprint compound. So while it doesn't have the carbonyl interaction, if it has got a hydrophobic interaction, then you might pick that up. So it's possibly worth adding that to the set of compounds that we're looking at to follow up on the benzimidazole. And on the right-hand side, we were actually, this was sort of inspired by looking at the cyclopropyl carbonyl on the blueprint. Although I've only shown one orientation, as I've already mentioned, the cyclopropyl could flip round the other way, with the carbonyl actually sitting in a different position. So you might, you could pick up potentially 2 different interactions with the lysine and the asparagine there. So that's the asparagine. The lysines over here, yeah. So possibly with having 2 carbonyls in that ring, you might be able. So some of the intermediates, these types of compounds are available. So if you're happy with that, we'll take a look at that as well.
Yeah. Yeah.
Okay.
I'm not sure how stable hydrolysis those are, but we can. Yeah, it's worth it to try. Yeah. Yeah.
Worth to see. Yeah. I mean, phthalamides, they're not ideal, but then we might be able to work around something else. We had a few other speculative ideas that looked a lot more synthetically, or a lot less synthetically accessible. So they're things that we might be able to think about in the future if we want to sort of, play around with these sorts of positions. So we're trying to sort of stay away from the mainstream of the main series, and see if there's any way of coming up with something that might ultimately generate us a second series.
Yeah, that's great, Sue. Yeah. Oh,
okay.
That's it.
That's
okay.
my lot.
That's fantastic. Yeah. Okay.
Hopefully we'll get some compounds out in the next month or so.
Great. And last but not least, David.
Okay, so no, I'm good. Okay. Just a little highlight about where we are in terms of the overall project. Just want to mention that we are now screening principally against ALK4 and ALK2, as well as, of course, ALK5. And what we've been finding, and this is actually a nice little outcome, is that when we're active on ALK2 and selective over ALK5, we're also selective over ALK4. So it seems that ALK4 and ALK5 travel together. So that's a nice outcome for us. If we don't hit ALK5, we know we'll likely be clean on ALK4. And also we're kind of, in terms of the last submission, we're at the 160 odd compound mark. And so now we're really trying to focus some of these series a little bit more and try and work around some of the things that we do know are beneficial. And that's, of course, principally this 4-position methyl substitution, and then trying to evolve other portions, principally the southern portion of 2009, to see if we can pick up some other favorable properties. So just a final word on the trimethoxy aryl portion of the molecule is that we've been trying a number of substitutions, deletions of O-methoxys, movements around the ring, additional substitutions. And this was one of the final examples we submitted with a shift of the O-methoxy. And the take-home message is that nothing is quite as good as the 3,4,5 trimethoxy, with perhaps the exception of this fluoro substituent on the 4-position methoxy, which does show reasonable ALK2 activity and also not too bad activity in the nanobread. So that might be a possibility if you want to explore it, but it doesn't, I think, add materially to where we're going. So at this point, we're ceasing work on the trimethoxy variants. And as Sue was discussing, the idea is then we can start looking at replacements entirely of the trimethoxy aryl. One of the things that we have been working on in terms of replacing that trimethoxy aryl altogether has been looking at these ortho-substituted amides. And the fluoroamide was one of the first hints that this might be possible. And we had actually good ALK2 activity. Unfortunately, this ALK2 activity is eroded by substitution of the 4-position with the methyl that we do employ in 2009. And we also see that methylation of the amide abolishes the activity altogether. So we've learned a few pieces of SAR here. Pleasantly surprised by the fact that when we substitute the fluoro for a methoxy, we do find that we have very potent compounds and they do preserve activity in the cell-based. But mind you, here we don't have the 4-position methyl, so that's in process of being made right now. So we'll see if it has an adverse or detrimental effect, as it did in this example right here with the fluoro. So yet to be determined, but this is a series that we're trying to build out a little bit more to see what's possible. Another thing that we've been looking at was just some very kind of different replacements of the trimethoxy. Just to highlight what a recent example here. With the sulfonamide, this compound unfortunately is entirely dead, so that's not something we'll be pursuing. But there was a case of this pyridone substitution here. This compound turned out to be decently active on ALK2, and preserve some cell-based activity. So one of the ideas we've been throwing around just yesterday was to see if perhaps we can design a modular synthesis such that we can explore a wide range of substitutions at the 4 position, ortho to the methoxy group, and first do so by virtual screen and then look to an outsourcing partner to see if they can do some quick chemistry to develop a fair number of compounds around this motif to see if we can perhaps hit on something else akin to this peridone with some better activity. So that's something we're going to be putting together in the next little while and hopefully execute on in the coming months.
David, 2137.
Yep.
So what do you think would happen if we introduced the 3-methoxy?
Right. It seems that the 3-methoxy does play an effect in potentiating these compounds. We could make that example, although I'm anticipating that given that we're greater than micromolar, we're not going to see that significant a drop to an area, to a potency where we can utilize, but we can certainly make that for completeness.
Yeah, I'm just curious because we have the primary amide in the methoxy amide, and that was pretty good, right?
Right.
Until maybe if we just make the para amide without the methoxy as well, and see how that compares as well, right?
Yep.
Just to kind of tie all the SAR together.
Yep.
It makes a nice story.
Yeah. No, absolutely. That should be straightforward.
Yeah.
Good.
Yeah.
So here was just a quick summary of starting to look at the Perazim modifications. We found that methylation doesn't adversely affect the potency, cell-based or otherwise. And then we saw that going from the piperazine to the piperidine was also tolerated, both unmethylated and methylated. And it was this piperidine that opened up some new possibilities. And so we started to play around that motif. And one of them was looking at the unsaturated compound. And this compound behaves quite nicely with good biochemical and cell-based potency. And then we thought, can we take this double bond and, of course, do some transformations and look at compounds with substitution at this position here. And as Zhang Fu mentioned, and when he showed his nanobret slide, the fluorine and the hydroxyl are tolerated at that position, and the cell-based values are quite good. The methoxy has been completed, and it's going to be screened in the next screening cycle, so we'll see what happens there. And this allows us, of course, to modulate some of the properties of the compound. So this is a nice little vector that we can potentially exploit if we see fit down the line. Just a quick note on substitutions around this central arrow ring. Just the take-home message is that so far they've all been detrimental, if not in the biochemical assay, then somewhat in the cell-based assay, with the exception of the orthochloro substituent. And now, as Sue mentioned, there might be potential to shift this nitrogen to this ortho position here. And so that's something we'll take a look at. We should be able to make that compound quite easily and see if there's anything to be had there. Bulking up this chloride to the CF3 group was a little bit detrimental, but still some activity preserved. So again, suggesting, as is common with a lot of kinase inhibitors, there might be some room to play at this position if we do indeed decide to take a look at that. There was also the southern portion explorations around the tetrahydroisoquinolines, which proved to be quite potent and also very good in the cell-based assay. Unfortunately here, addition of the 4th position methyl group is a little bit detrimental, both in terms of biochemical and cell-based potency. But again, this is something, it keeps it in line essentially with 2009. And this is something we might be able to work around if we do some more judicious substitutions around that tetrahydroisoquinoline. This phenomenon of reducing the activity with the 4th position methyl is also held in this other example where we've shifted the nitrogen in the isoquinoline. Right now, ongoing is screening of this compound, which was just made recently to see if this southern moiety has any impact or is amenable to a substitution with this methoxy amide. So this remains to be seen whether these two work together.
Yeah, that'll be an interesting compound.
Yeah, and if this is, in fact is active, then of course you can imagine we'll start. We'll put the 4th position substituent in and start looking at playing with that series. Just a little kind of grab bag of odds and ends for the south here. Many of which these first two you've seen last time. We tried this bridged piperazine, again potent and biochemical, a little less so in cell-based. So not really something that we're looking at too much, but we can look at some of the other isomers, and we do have a couple things in plan around substitutions on this ring. So we'll just kind of tidy that SAR up, but this has not been beneficial. This is also this Novartis southern piece. Again, recently made compound. This should be in the next screening cycle, so we'll see how this plays in our 2009 core series. And finally, we've been, of course, looking at the 4-position substituents outside of the methyl group. And one of the initial ideas we had was that perhaps an electron donating group would be beneficial for the H-bond acceptor characteristics of this nitrogen. In fact, it turns out that we got it entirely wrong. And it seems that, in fact, electron withdrawing group, at least in the N of 1 example that we have so far, seems to be beneficial. And this nitrile is turning out to be quite an interesting compound because it's both potent in biochemical and very potent in the cell-based. And as I'll touch on in the next slide, it also has slightly different properties, which we might find of interest in terms of the brain tissue binding and whatnot. Recently just made the CF3, so this is in the next screening cycle. So we'll see if, again, an electron withdrawing group is equally beneficial or as beneficial as the nitrile is turning out to be. And of course, for the purposes of SAR completeness, I'm working on the fluoro at that position. So we'll see what happens there. And at that point, I think we'll have covered the kind of lion's share of the groups at that position, at least ones that we would find have utility going forward. And just a quick summary slide. If you go to any kind of literature review on drugs for the CNS, you'll find that there's a kind of general agreement in what kind of properties are favorable. And so this is just from one somewhat recent review, some of the characteristics that we're after. And you'll notice that wherever we have a green check mark is a characteristic that we do in fact abide by for 2009. The one kind of, which I put in yellow, is metabolic stability. For 2009 in our MLM and HLM, we're a little bit lower, like 70-odd%, but really fundamentally no difference because ultimately our PK results were quite good. So we have green check marks across the board. Where we don't have a check mark is data we don't have a direct comparator for, but some of these pieces may fall into play down the line if we do choose to get that data. Just to highlight here at the bottom of this little kind of properties rundown for 2009, we have some results for plasma protein binding. We found that 2009 is about 6-7% free in human, and in mouse, in our hands, it was about 9% free, or rather, I think this is the CRL result, excuse me. And then there's a result which I think Jeff will elaborate a little bit more on, courtesy of Sue and CRL, which is the measurement of the mouse brain tissue binding, and we found that we're about 1% free. So this 1% free has implications in terms of the free concentration based on our PK results at the 2-hour and 4-hour time points in the brain. And so the suggestion here is that at a concentration of 55 nanomolar after 2 hours, we are around or above, depending on how we measure, how we interpret the nanobret result, the EC50 of the compound at that time point. Whereas in 4 hours, we do start to fall beneath it. So one of the ideas is that we would like to perhaps reduce the brain tissue binding by some manner to see if we can increase these figures a little bit. And one of the ways that we're contemplating doing so is to take a look at reducing the lipophilicity of these compounds, because if you do take a look at 2009, the log P is quite high, around 3.3, and PSA around 55, which is quite low. So if you look across, I just put some representative examples of that series here to show you how, if we do in fact decrease the log P by introducing, for example, the nitrile, we do increase the PSA. And we have a range of possibilities in terms of the southern substituent, in terms of these substitutions here on these piperidines, as I discussed. So we do have the option of tuning the polar surface area and the log P, should we be interested in looking at some of these compounds in the brain tissue binding. In terms of the other series, you can see that we are considerably more polar once we do introduce the amide. This may be detrimental or it may not be going forward. So that again is something we can look at. Because you can see here, the C-log Ps are considerably lower. But interestingly enough, if you look at CNS drugs in aggregate, the average C-log P does actually tend to be about 2.5. So, you know, we might have some possibility of getting away with this if we do choose to look at it in the future. So that's where everything stands right now. So we continue to make compounds. principally around the 2009 series, exploring some of the different trimethoxy replacements, and now potentially looking at tuning the southern portion for polar surface area and whatnot. So that's it.
I do like this whole dimethoxy compound that you mentioned earlier. Give a little bit of potency.
Right. This guy right here.
Yes. I mean, have you had a look at PK and the protein binding those compounds? Because
yeah, now we're saying the fluorinated dimethoxy system compared to the trimethoxy. I'm wondering if that offers you some metabolic advantages and stuff like that, and whether or not the binding and tissue binding properties that you're after. So you might want to go back and gather some more data for that.
Yeah, that's a good suggestion. Okay. I mean, we haven't looked at that compound yet, but you know, I think certainly making it with the 4-methyl
comparative
as well is probably worthwhile. We can improve the selectivity a little bit, and then
One recommendation. I mean, a lot of your lead compounds, you should actually explore the trimethoxy and that, just to see what they offer you. Right. Yeah.
Yeah, that's maybe another one we can add to that kind of permutation matrix that we were talking about, right?
Yeah, I do like that compound a bit.
Good. Okay. Thanks, David. Great update and great progress, I think. Okay. So again, I just wanted to remind everyone about the CTIP lead profile. I presented this at the last meeting, so it's really just here for reference. We did select several compounds to move into a brain exposure experiment, and M4K 2009, as I presented at the last meeting, really showed good brain exposure and brain to plasma ratio of about 0.3 to 0.4, 0.5. And so it's really, we think, our most promising compound that we've identified to date. And so the next steps were really to check the brain unbound fraction versus the plasma unbound fraction, which is kind of an accepted, if not somewhat contentious, way of evaluating how effective a potential drug will be in the brain. And so we did that, and we also wanted to further profile the compound against a variety of kinases. So I'll just re-show the data so that everyone's on the same page for the rest of the meeting in terms of getting into the discussion. So we did profile M4K 2009 at RBC against 374 kinases at 1 micromolar, and we only hit 10 of those at greater than 50% inhibition. ABL was 1. This is one that we also saw with the original compound 1055 as well. DDR1, this is one that we hit actually pretty strong. Now you can tell that we hit ALK 1, 2, and 6, which is not unexpected. We already knew that, so it's good that they came out in this, but none of these are anything really to be that concerned about. DDR1, we hit essentially as hard as we hit ALK 2, which is, I think, interesting. And Alex, as we were talking about earlier in the meeting, has been involved with this kinase before. In fact, he's published on it with an interesting class of compounds. And this was to target inflammation, I believe, right, Alex, after a lung injury?
Yeah, I think so. Yeah.
Yeah. So, you know, I don't think that this is anything to be that concerned about in terms of hitting this, but it is something that we will follow up on and get full IC50s for this kinase. These other three, we don't hit that strongly, but again, we'll add them to the list of kinases that we evaluate M4K 2009 for a full IC50 curves. TNIC as well is one that we hit again pretty hard and we need to follow up on. This is involved in Wnt signaling and has been targeted previously as a potential target in colon cancer. So, you know, again, this is one that we need to get an IC50 for before we worry about it too much. But it's, I think, interesting to see what other kinases we're hitting with this scaffold. And it'll be interesting, I think, as Alex mentioned as well. So what's the impact of that C4-methyl on these other kinases, and do we hit these same kinases with some of our other compounds? And so I think that over the next month or two, our output is going to decrease a little bit in terms of evaluating compounds against our standard ALK4, ALK5, and ALK2. So I think, we can start to profile some of our compounds against some of these other kinases as well with our RBC friends. So I should also mention that David Drury has sent this compound for the full kinome profiling at DiscoverX as well. And so it'll be interesting to compare those results with the different assay formats. And, we'll see when the results come in, how those results compare. So overall, though, I think, hitting only 10 out of 374 kinases, in which 3 of them we already knew about, and really only these 2, I think, very strongly, is quite promising. And so this is a quite selective kinase inhibitor. The M4K 2009 versus the TPP looked really quite good. We're as potent as we think we need. The metabolic stability is good. CACO2, the beta-A is acceptable. CYP inhibition, Sue presented this at the last meeting, that we didn't really see anything against 5 major isoforms, so it doesn't inhibit CYPs. We've sent the compound for hERG inhibition, and we're anticipating those results very shortly. Compound will be sent with the contract today for Ames, so we'll evaluate that. The in vivo profile of M4K 2009 looks very good. The brain to plasma ratio, the target of greater than 1, we talked about this at the last meeting. I think that's hopeful and wishful thinking, but I think, the 0.4 to 0.5 that we're seeing with M4K 2009 still represents, I think, a very good exposure in the brain. And then the key piece of data we were missing was the unbound brain fraction. And they're targeting more than 3-fold the DIPG CC50, so we don't actually have any DIPG CC50s at this point. So I think I'll show you a slide where we're using the nanobret data as kind of a surrogate for this. And then in vivo efficacy, obviously, we have not ran yet. So Sue and her friends at Charles River were able to run the mouse human protein binding, and as well as the brain tissue binding experiments for this set of compounds: 1062, 1055 with the 2-methyl, 2009 with the 4-methyl, 2045 with the 4-methyl with this N-isopropyl, and then the M4K 3007 compound that comes from the Blueprint patent. So the plasma protein binding numbers were pretty reasonable, we thought, for all of these compounds, but let's just focus on M4K 2009, because really that's what the rest of, I think, our discussion should be about. Also, I think 3007, and I'll get to that. But so the mouse plasma protein binding was 91%, the human was 97%, and this number in brackets is the data that we generated here at OICR for the human plasma protein, and it's within the same ballpark of what was generated at Charles River, but there is some variability, as you can see. The brain tissue binding, all of the compounds were highly bound, including M4K 2009 at 99%. This is, I think, higher than with what we were hoping to see. It does have implications, as David showed in his presentation, and I'll show the slide that Sue generated on, again, in my presentation, but, it is what it is, and I think that the exposures that we're generating with this compound, I think it should be suitable for a proof of concept compound. I don't know if anyone disagrees with that, but again, keep in mind that we've only dosed at 10 mg per kg so far. So when we dose escalate a little bit, and you'll see that that's part of the plan, is to see whether or not we can get above at least, and stay above the EC50 in our nanobread assay with this compound for more than the 2 hours that we see now at 10 mg per kg. I think it's interesting. Yeah.
Sorry, I was just going to say, I think you're absolutely right. I mean, even though, it does seem to be highly bound, I think you've still got, because you've got very good PK profile, that does leave you with, a significant amount still getting into the brain out to a little while. Not perhaps as long as we might have liked, but I think it still looks it's disappointing, but it's still reasonably promising.
Yeah, but this is again, as Jeff just mentioned, this is just one dose. We didn't do any dose escalation or dose linearity, and those kind of things what we have to do is go 3 times higher than the EC50 we're aiming for, right? So it should be.
Yeah, I think it should be feasible. It's just how long can we maintain it, and how long do we need to maintain it to have an impact on the disease, right? That's still some questions that we just don't have the answer for. But, compounds like this will help us, get that information. I think that's really what we've set out to do here is, generate a proof of concept compound as quick as possible and try to generate some in vivo data that gives us the incentive to move forward. And who knows? I mean, 2009 might even be a clinical compound, right? So this is the only wart that we've found in the compound so far. So I think things continue to look good. I do think M4K 3007 looks really quite interesting. I think it's slightly lower bound to the brain tissue. So, that's interesting.
What's the fold above EC50?
We don't have PK for this compound yet. So we don't know. We don't have PK or brain exposure. And so that's part of what I think should be a future plan, and something that I'll show is that maybe this is a compound that we move into some in vivo experiments as well, because it is quite potent and quite selective, even more so than I think M4K 2009, at least against ALK4 and ALK5. We don't know about the rest of the kinome, and that'll be data that we'll have to generate as well if we're serious about this compound. And, anyway, I'll get to that in a future plan slide, because I think there are some caveats around that, but I think it is an interesting molecule for this reason.
So one question, just to make sure that I'm clear, this is Javad, this is Children's. The way that you're looking at the brain-bound molecules is that the mice are fed with a compound, and then the brain sample is collected and analyzed. Is that how you're looking at this, right?
Sue, do you want to answer that?
Yeah. So the brain exposure experiments that are done there, that's where you do effectively a PK. So you feed the animals and then take plasma and brain and do the compound concentration. So that's how you get the exposures. So these are looking at how strongly the compounds are bound to protein, either in just sort of generically bound protein in either the plasma or the tissue or the brain tissue. So these are in vitro experiments, the tissue binding experiments.
Right, it's a microdialysis.
Yeah, microdialysis tissue. It just uses tissue, not animals.
I see. Okay, great. So as a part of our work, we will be doing actually this in vivo. And I think one of the most important questions to answer is that once and when the compound enters the brain, how long it would linger in the brain. And that's an experiment that we have, and we could answer some of these questions, because we would have live animals with the probe in the brain, and we can collect serial samples and look at exactly how much of the compound is in there and how long it will stay.
Right. I think, just to step aside here. So, yeah, Javad, I think, this kind of goes to the kind of free drug hypothesis, that drug that is bound to either protein or your target tissue isn't available to bind your target. And so that in some cases, and in many cases, I would venture to say that does pan out, that, you know, your free drug does correlate better with your observed efficacy or your observed pharmacodynamic effect than absolute drug concentrations, but it's not always the case. There's a lot of variables in terms of how strong it's bound, how strongly it's bound, what that equilibrium looks like, and, you know, how strongly you actually bind the target as well, and how long you stay on you know, the residence time on target as well is also important.
That's really important. Yes.
Right.
Right.
So you know, it's a number. It's, you know, obviously we'd be happier if this were, you know, lower. But you know, I think that, you know, it shouldn't be a showstopper at this point for this compound. I think we can still forge ahead and try to generate more data to try to understand whether or not that hypothesis, that free drug hypothesis, is relevant for this target in this indication.
Sure. That makes sense. Right. Thank you.
Yeah, one question. Have you done, so you showed the broad selectivity profiling against the kinases. Have you looked at CNS targets?
So we have a SERAT panel that's ongoing right now.
Okay.
In fact, I heard that it's done and the report has been released, but it was released to Owen. Okay. And Owen's on vacation, so I haven't had access to it yet.
Okay.
Oh, they released it to the person paying the bill. So, but it was the safety screen, 44.
Oh, yeah.
That we use. So there are some CNS targets in that.
Okay.
So we should have that data any day. So yeah, this goes back to talking about that free drug hypothesis again, and David already showed this slide that Sue generated, talking about the free concentration of M4K 2009 at various time points based on the pharmacokinetics at 10 mg per kg that we have so far, and based on the nanobret EC50s. And you can see that at 2 hours, the free drug concentration after a 10 mg per kg dose is around the EC50. So, you know, we're in the ballpark, we're in the game, and you know, some very subtle changes, either increasing dose or if that plasma or brain protein binding is just, you know, 1.5% free, right? You know, you could imagine that we could be above it for quite a bit longer. So
I mean, this is just a snapshot based on the exposure you've done with 10 mg, but I think ideally you might want to do an MTD first.
Yeah.
And then when you get to an MTD, you actually repeat this experiment to see, you know, how high above, you know, your EC50.
Right. So that's part of the plan. Yeah. So before we get to the plan, yep.
Can I just ask a quick question? So when you did the permeability assay, you didn't see, there was no efflux observed, was there?
No, for 2009, no.
Yeah.
No, it was. I took KCO2 off of this, but no, it has an efflux ratio of you know, it's essentially one.
One or two, yeah. So if it is not above 10, we don't worry about those.
Yeah, I think I have it. I have it on another slide, I believe.
Oh, so that's okay. I was just, because it's unusual to see such a low brain to plasma ratio, unbound brain to plasma ratio, when you've not got any significant efflux, particularly for pretty lipophilic compounds. It's normally the opposite. So I think the results are quite unusual.
Yeah, the other thing that I did notice when they ran the brain tumor, or when they ran the brain exposure experiment, was that the plasma exposure was actually about twofold higher than what we had seen in our pharmacokinetic experiment. So, you know, I think that there's still we need more in vivo data with this compound to get more N values, you know, to get a better real understanding as to where we are.
As I think I mentioned last time, from talking to our in vivo guys, we would have quite concerns about taking 8 time points by serial bleed from mice, because you can get some skewing of results, because by the end they're down to pretty low amounts of blood they've got left. So you're taking quite a lot of relative proportion of blood from a mouse when you do a serial bleed. That's why we often tend to do the sort of more, we do terminal bleed. So you have to have more mice, it costs more, but I think you can get a more realistic indication of plasma and brain concentrations. Just
right. Yeah, no,
just something
that
we're
was
more familiar with doing.
I'll go to another species. Make it easier.
Yeah, that was something that you know, I think we talked a little bit about it at the last meeting. We didn't really have a chance to discuss it, and you know, I think it would be interesting to move 2009 through the series of experiments that you guys have recommended, and to see how our results compare. You know, at this point, you know, the recommendation to kind of move earlier compounds into that full battery of experiments at risk is something we just don't have the cash to be able to do that.
I understand that. Don't worry.
Yeah, so I think with new compounds, we'll kind of move forward with the path that we've already done. Get the PK first, make sure that we're on board with reasonable pharmacokinetic properties, and then move into the brain. But the more advanced compounds, I think, you know, it would be great if we could, you know, get you guys on board and do the series of experiments that you recommended at the last meeting. So the M4K 2009 certainly is one that we would like to move into that series of experiments to get more reliable data than I think we have now. I think the data we have is encouraging, but, you know, there are certainly some questions around it, and, you know, perhaps the serial bleeding is to blame. So I think it would be a good idea. And it doesn't hurt to get, you know, the brain to plasma ratio in via a couple of different ways as well, right? So, yeah.
And I think too, I mean, eventually going across species, I mean, people have had surprises across species where you see good brain penetration in mouse, and then you go to rat and monkey, and then it's really bad. So, I mean, there's still quite an uphill battle in terms of just fully evaluating penetration potential.
Yeah, there's a lot to do still. You know, I think the goal for now is to make sure that we have a compound that we can do proof of concept with in mouse. Exactly. And if it continues to look promising, then we'll move to the higher species. Yeah. Okay. So not to go back to the SAR, but there was a couple of, I think, interesting SAR points. So M4K 2009 introduces the C4 methyl, has the NH at this position, and it's, you know, we all know how good of a compound that is. Interestingly, N-methylating that compound really results in a metabolic liability in the liver microsomes, whereas in other series, we don't see that. For example, going from the NH to the N-methyl, in this series of compounds, we see a small decrease in the metabolic stability, but in this series, it really is substantial. So interesting, but you know, and I think when we had the isopropyl here, it was fine. So there are ways of substituting that nitrogen and staying away from the metabolic liability, but this compound just resulted in a very rapid oxidation and probably elimination to generate.
That's why I say if the metabolite is 2009.
Well, we can measure that, right?
Yeah.
Oh yeah, we can check it. Yeah, we've got both compounds, and yeah. And then the other interesting point on this slide, I think, is so M4K 2043. This was a compound that we thought was very interesting. We moved it into mouse PK, both IV and PO. It had pretty good bioavailability and you know, essentially similar pharmacokinetics to M4K 2009. However, this compound did not very well penetrate the brain. And this compound does show a moderate efflux here of about 2 to 3-fold. So, you know, we thought that was an interesting result, and potentially something that we can track to look at this, whether or not we're going to have good brain penetration. But when you N-methylate this compound, now you're reasonably metabolically stable. You're essentially equipotent. We don't have the ALK5 data yet, but now we've kind of dealt with that liability of the efflux. So it's possible that a compound like this now might have better brain penetration than 2043. It might not be related to this nitrogen, which is different between 2009 and 2043. So I think that that's something interesting that we can follow up on as well. The promising new compounds to profile. I think David did a great job highlighting some of these. Again, 2112, potent, good selectivity. We at least in the biochemical assay. And Zhongfu, these are some of the compounds that I know that you've highlighted to get the ALK5 luciferase data for. So 2118 as well, I think is also interesting. Interestingly, it is metabolically stable as well. The fluoro and the hydroxy analogs that David presented, the newer analogs in his presentation, I thought were also very interesting. Shows that you can substitute at this position. This one, though, I think is very interesting just because of that huge selectivity window that we've opened up. And like I say, it is metabolically stable, so I think it's an interesting molecule. These tetrahydroisoquinolines, I think, are also, very interesting, very potent. Unfortunately, introduction of the C4-methyl did result in a decrease in potency when we had this, but there's a possibility, and I think David showed in his presentation now with the nitrile, that we can potentially improve the potency of these. So making some combinations of these things to try to build in the best properties and the best potency, I think could lead to some very interesting compounds. And then, as we said, the nitrile and this methoxy amide, I think, are also very interesting observations that we're going to continue to follow up on in terms of the follow-up SAR. So again, doing permutation matrices around some of these key findings. I think adding in the dimethoxy fluoro in terms of the top, these are also things that we can do where, these can be very potent, the nitriles improve the potency, but, I think there's still a lot of data to generate and, especially the ALK5 data for some of the compounds to make sure that we are selective or improving selectivity. So the next steps, and after this, I think we'll kind of move into some discussion about this, and I'm certainly open to alternative suggestions. But we're looking at scaling up M4K 2009 to about a 10-gram scale to make sure that anyone who wants the compound, anyone who wants to test the compound in whatever assays, it will be available for them, and as well for us to continue profiling it as well. The HERG, Ames, and SERAP are all in progress or will be very shortly. The Ames is the compound for Ames will be shipped out today. IC50s for the off-target kinases, we need to schedule that, but this is something that we want to have done hopefully before the next meeting. So we'll get that going. Evaluate versus a panel of DIPG cell lines. I think, Javad, this is something that you mentioned you can do. I know that Jerome is also evaluating some of the compounds against the cell lines that he has, and it'll be interesting to see, how potent we are and how the compounds behave against a variety of different cell lines. Now, I think,
I was just going to interrupt briefly.
Yeah.
So Zhongfu just did, and Lita just did a few viability experiments, sort of exploring setting this up. 2009 actually looked quite good, so we're certainly as good as anything we've tested in the past. So
in which assay was that? Sorry.
Both in a cell titer glow and a PI staining, an Nexin staining for apoptosis, and other assays.
Oh, okay. So it was good. Okay, that's great. Yeah.
We don't necessarily break new ground, but we're certainly as good as anything we've screened before.
Perfect. That's great.
Hopefully we can maybe have some slides for the next meeting.
Okay, that'd be great. Yeah. No, but that sounds encouraging. That's for sure. I also know that Alexi was going to look at some MTT, some cytotoxicity assays with some of our compounds as well. So, I think the more data we generate, the better. I think the in vivo portion, especially with respect to efficacy, is still something that we need to finalize as a team. I think I very briefly mentioned this at the last meeting, that it might be interesting to look at our compounds in an FOP model. I think some of those models are more validated than the currently available DIPG models. I mean, ultimately, we need to show efficacy in a DIPG model, but getting some of the PK/PD and safety data and dose response in a proven FOP model might be something that will give us more confidence moving forward in terms of, how much compound do we need to have on board to at least affect that disease and give us a target when we do move into the DIPG in vivo model. So I think that's still a discussion that I'd like to have, and I put that again on the next slide. That's kind of lays the groundwork for the rest of the meeting in terms of discussion, and I'll be interested to hear other people's opinions. And I'm not sure, Sue, if Martin is on this call, but I'd certainly value his opinion on that approach as well.
He wasn't able to make it today, I'm afraid, but I will certainly have a word with him when I see him next.
I think it's something maybe we can schedule a call as well.
I'll try and sort some. Do you want me to try and organise something?
That'd be great. Yeah, sure, please. Yeah.
I mean, one recommendation. So even though we like 209 a lot in terms of its properties, with some of the discussion we had earlier with the NH and that variability in PK and stuff, I'm wondering if it's worth doing a lot of your preclinical, looking at both the N-methyl and the NH, so that at least you have that comparative data, particularly for a lot of the permeability and CNS penetration. So if you're getting more consistent data with the N-methyl, just to be able to make it, is it the NH and the liability of being a PGP substrate or something like that. So
you mean like dosing the N-methyl?
Yeah, like at the same, every study you do with 209, you should at least compare the N-methyl, because I think the stability was bad in humans, but I think for your proof of concept, it was about 30–40% in mouse. It might be good enough to kind of say, even though you might get poor exposure, does the partitioning and permeability profile overall look a lot better, to kind of just better understand, brain penetration property. So, for some of the early studies, whether or not you just compare them alongside each other until eventually you get comfortable and say, hey, you know what, 209 is just as good, and it has more favorable profile in human going forward.
Yeah, I think, we had the isopropyl analog. That we did move in. In fact, it was in this experiment as well, M4K 2045.
Okay. That would help. That would answer that question.
Yeah, and do I have that slide? Brain exposure 2045. So it showed good brain exposure as well, and that one is metabolically stable. So I think it's so we did move that through. We didn't further profile it in the kinome and in the HERG and the Ames, but it does partition to the brain.
Yes.
And
how hard would it be with brain tissue binding on that compound?
How high is we have the data. 2045 was 99% as well.
99%.
Yeah, it doesn't offer any advantage there with respect to 2009, and but the exposure was a little bit lower, although the ratio, right, brain to plasma for this compound was actually better. But it cut by half after 2 hours. So we were wondering, from between 2 hours and 4 hours, that ratio dropped by twofold. So is it being selectively metabolized in the brain? Or I don't know.
To get that, I think you need a full profile to be able to be sure of that. Full brain profile over a period of time. I guess I think David mentioned it during his presentation. Part of the issue, I think, with these compounds with the high tissue binding is is the log P. They are pretty lipophilic molecules, so I think that doesn't help us.
Yeah, and as David showed as well, I think very nicely. Some of the modifications we're making now might address that. Again, if it's important, if it's important, right?
I think that's where you have to answer.
Exactly. Yeah, and I think
push the compound, push it hard and fast.
Yeah.
Get into the FOP model. I think that's easier to run.
Yeah.
See if it works.
Yeah.
And then regarding your list, I noticed you're doing different PK, but you might even just skip the PK and do the MTD at different doses.
Yes.
And then measure the concentrations there.
Yeah, we could do it that way too. Yeah, I thought about getting a full PK profile at both 25 and 50, to try to push it, but, a full MTD, I guess that's something that we can discuss internally. I don't know if we'll be able to run it here or not, but given...
If you want to have a call to discuss some of these PK things, I can set something up with our DMPK guys. Yeah,
that'd be great, Sue. I think we should do that, and really, I think this list is interesting. We have a little bit of time, while we're scaling up the compounds and everything, to be able to decide exactly how we want to move forward. But I think getting these things scheduled, especially if we do want to move into an FOP model, we need to identify a lab that's capable and willing to do it. And then, certainly, I know Javad has some interesting models for the DIPG, and Chris Jones as well, and that group has the DIPG 007 model, and I know they're willing to help, and so that's fantastic, and we look forward to that. But I want to know that we're able to impact a disease that's relevant to ALK2 with our molecule, so that when we move into these models, we don't have the question: is our molecule good enough? Is it is it the molecule? Is it the model? Or is it the hypothesis? And if we can eliminate at least that it's not that the molecule is fine, then I think getting that answer from the FOP models could be interesting.
And I guess the question is, if it didn't work in the FOP model, would you not take it into the brain?
I probably would not. No, because it's the same target, right?
Yeah.
It's the same mutations essentially. There's several interesting models. I have a slide on that as we move forward.
Just a quick note, Jeff. Just to point out, like MTD and PK, these are good ideas, but the one thing one has to keep in mind is the compound requirement, right? So if you go for an MTD, a multiple compound, you need substantial amount of those compounds. So
that's why we're scaling it up to 10 grams.
But one compound is fine. But like if you want to do multiple, I'm talking about. If you want to do multiple compound, doing MTD without skipping the PK, that's the point.
Yeah, I think one suggestion would be like, David, if we pick one compound and we do an MTD, and we realize, okay, we can go up to let's say 100 mg per kg, you can then decide, okay, I'm going to fix 50 mg per kg dose.
Yeah.
And then that becomes the benchmark for all of the subsequent compounds. I mean, you can decide, hey.
Everything is speculation.
Exactly.
Yeah,
exactly.
That's if we fix 209.
Yeah, we've tested everything so far at 10 mg per kg because this series has pretty good PK even at that dose, right?
Yeah.
So if you go too high, then you might start saturating something. So I didn't, or causing deleterious effects as well, right? So I didn't want that complication in our PK experiments, but certainly I think with 2009 we can start considering the MTD and maybe both, right? I mean, we'll get the same data, so 25, 50, and we'll have to determine how long we want to do the MTD for. So I think that there's some
5-day MTD.
Yeah, that's usually what we do here, but but I think scheduling the call with Martin and the DMPK folks at CRL would be a great idea. Yeah. and we can all discuss this and make sure that we're all aligned as a team to move forward. So yeah, I think as well, profiling the M4K 2009 analogs, that's moving along. The in vitro ADME, the PK of the newer analogs, we talked about this through the Charles River versus Farmeron, and I think I'm in favor of, at least for new compounds, continuing to profile them the way we have, kind of stepwise, cheaper. It's
fine.
Yeah. But for more advanced compounds, I think moving them into the battery at Charles River is something that we should do. So, and then Alex briefly mentioned that there's some work ongoing in terms of trying to develop an ALK5 nanobret tracer. So David Smeal is working on that, and we're going to try to make one or two in-house and outsource the rest. But that I'm requesting quotes now, so we'll see how long that'll take. I think it'll move faster if we do it that way. And then, of course, continuing the SAR, improve the potency selectivity in this unbound brain to unbound plasma ratio, replacing the trimethoxy phenyl. I think methoxy amide permutations that we talked about, trying to lower log D. I think that's a really good idea. And we'll have M4K 2009. That's a kind of a high binder, and if we can identify another compound that has a better ratio there, and move that through a similar battery of experiments, we'll be able to tell how important that actually is for in these models. So, and then as we mentioned earlier, this M4K 3007 I think is an interesting compound. We could also pursue that as a potential proof of concept molecule. And I think Sue will have to talk maybe offline or about how much of that compound is available and how tough it will be to scale up. I don't think that there was that much available. So
no, we're talking in the 10s or 20s of mgs at most, I think, of that compound. I think when I looked it up last. So, and it's not the most trivial of syntheses. It's not quite as trivial as some of the other compounds, but we've made it once. We can make it again.
Right.
Or if you want to outsource it, that's always an option.
Yeah, I think getting some of the data with the material that we do have in hand, might be a reasonable approach. At least get the pharmacokinetics, because if we don't see anything with it, then it's done, right? So I think at least get that first. So we might be requesting you to ship some of that compound, some more of that compound. Okay, so with that, I think we have about half an hour left. I think we've had good discussions as we've moved through all of these topics. I think the FOP model is certainly something that I'm in favor of at least trying to identify a lab and that's capable and willing to do it. I know that there are several validated models out there that do work in the presence of compound. Michael Franti from BI, when he presented their FOP project, highlighted a couple of these models, and I know Alex is involved in an FOP project that, so there are, I think, some good models out there available that will allow us to establish the PK/PD, maybe get an early read on a safety signal. I know that there's heat shock inducible adult zebrafish models as well, which might be interesting in terms of looking at a higher throughput experiments that could allow us to really prioritize compounds quicker. And then the model that Michael Franti showed us from Dr. Levi at University of Michigan might be an option as well. So, I think this is something that I'd like to explore more thoroughly. I think it could give us some very valuable information about our compounds that will help us design the in vivo efficacy in a DIPG model study.
So my feeling, Jeff, is that your compounds will work tremendously well in all of these assays. The only thing you would learn is maybe reducing the dose, or are you thinking about safety?
A little bit of both, I think. So I expect they should work as well, based on, I think even 214, 117 Michael Franti showed that one worked pretty well in this HO model. But what I'm more interested in is dose response. How high do we need to be, right, to elicit a maximal pharmacokinetic or pharmacodynamic effect in those experiments? And because presumably, that will give us an idea about target occupancy, and where we need to be in a brain exposure experiment to have confidence that we should see efficacy. So that's more along the lines. I expect them to work, but it's more about the dose response and the pharmacodynamic effect that we see.
So is there
okay, I'm happy to reach out to half a dozen FOP labs or labs in that space, both zebrafish and mouse, and see if we get someone who bites and would be interested to work with us.
So that'd be great.
I mean, I'm sure people would be interested. We obviously have to tuck into people who have the capacity, time to do it. So, but
yeah, no, exactly.
I can make a list and then run it by you, like suggestions of actually what we're proposing, and then we can send them out.
Okay, I think that sounds like a great plan. I think, we can, as well, I think when we set up this meeting with the Charles River DMPK folks, we can discuss it further there and further refine the kind of what we'd like to see in those experiments and what information we think we can get out of them. And I think that'll help us explain our case to your contacts.
Okay.
I'll try. Okay, if I try and set something up for next week, then, assuming the relevant people are around.
Okay, that sounds good. I'm around next week. Ahmed, you too? Yeah?
Next week, but yeah, I can probably call you.
Okay. Yeah, advancing.
If I send some potential dates to you, Jeff, then you can share with all the relevant people, other project team people that need to be present.
Perfect. Yeah, that sounds great.
Okay.
Yeah. So from what I've heard today, everyone is in favor of continuing to advance M4K 2009. So I think that's good. If anyone has anything that they'd like to say around that, please do. And then the M4K 3007 as a potential proof of concept compound. I think let's at least get the pharmacokinetics for it to see where we stand, and we can make decisions based on that profile. And of course, with that, we will have a freedom to operate issue, but at least with respect to demonstrating a proof of concept, I think we should be okay. Okay. Does anyone have anything else? Any other business? Any comments on any of these points?
I just had a comment, Jeff. With your kinome profiling, you were looking at ALK4 and ALK5 as off-target kinases, and you mentioned that ALK4 and ALK5 are very similar. I mean, we've seen that quite a lot as well. Is it worth skipping the ALK4 and changing that to ALK3 or ALK6?
It could be, although I think ALK1, 2, and 6 are pretty similar, right? And I think we did see that in the kinase profiling, that we do hit ALK6 at roughly about the same fold. So I mean, at one point we were looking at ALK1 as well, and they seem to be tracking pretty closely. The ALK1, ALK2, and ALK6. They're all these BMP.
Yeah, I think ALK1 and ALK2 track quite closely, but the ALK3 and ALK6, you can definitely make compounds that are selective there. More so, well, they're all going to be less than ALK5, but there's certainly scope to push those. And you can use the blueprint as the 307 or 3007 as a sort of control compound there, because I think that data is published, or not published, but should be available.
Okay, yeah.
I guess the issue with was the reason we switched to ALK4, because there was, I can't remember what the detail of what the concern was, but there was negative implications if you do hit ALK4. that right?
Right. It was also a cardiovascular
That was it, yeah.
issues that arise if you hit ALK4. That came from BI, right? From Michael Franti's presentation. So, you know, I don't know, Alex, are you worried that, you know, hitting ALK6 might be detrimental to, you know, to giving a drug to a human?
Not especially in this context. It's less well explored. The tissue expression of ALK6 is quite limited, I think, compared to ALK3, the other BMP type 1 receptor. ALK6 is quite heavily linked with skeletal bone development, I think.
Okay.
There have been knockout mouse, but yeah, I'm not overtly worried. I think you're right. ALK4 and ALK5 would be the chief worry.
I guess if there's skeletal issues, then you might need to be a bit more concerned if you're giving it to kids.
Yeah, I guess it depends on how long we're going to have to give the drug too, right? You know, do we give it, to treat and cure DIPG, and then, you know, you can stop treatment, or is this going to be a chronic drug? I think, again, that's something that we just don't know at this point.
Yeah, in those studies, you know, keeping an eye on ALK6 might be worthwhile, and we can also look at what's known about it.
Yeah. Okay. Yeah. I can take a look at that, and I mean, I know RBC has it, and we can. So that part of the RBC arrangement is that they will test a smaller subset of compounds relatively frequently against 5 kinases. So we can certainly add that in as one that we explore for more promising compounds.
We can do it in the nanobreath as well.
Oh, well, that's even better then, yeah.
I'll request that in the next cycle, which is next Monday. We send out for screening. So I can request 3 and 6 for, let's say, a handful of compounds.
Yeah.
Take a look at.
Yeah, it can't hurt. And yeah, okay. All right. No, I think that sounds like.
So we've not looked at any of the mutants recently, have we?
No, we haven't tested them, but 2009 was tested.
Yes.
Against the mutants.
Right, okay.
Yeah.
Okay.
Yeah.
Okay.
Alex, did you have another point?
I think at some point we were interested with David Drury to maybe send a couple of compounds as nanobreath tracers for ALK5, just to confirm they had potent binding to ALK5.
Right.
They're probably not, you know, they wouldn't be synthesized yet, so
no.
In the future batch. Right.
Yeah. I think David, maybe you can comment. I think you're planning on having it ready by the end of.
You're talking about 1046, Alex? Or you're talking?
Well, the nanobreath.
You're talking about the whole tracer. You're talking about Right. So I've already scaled up 1046. So I'll be shipping that to you on Monday. And I plan on making a couple of the linkers in-house here. I hope to have one also ready to go to RBC on Monday, but I'm not quite sure because I have some other stuff to do. But the intent is that I'll make a couple of the linkers here. We'll hopefully outsource a couple of the linkers also. And that I anticipate, well, on my end, they should be done within a couple of weeks. The outsourcing, I don't know yet. Right. But I was thinking wait till they all come in, collate them, and then we can send them for screening.
That sounds great.
Yeah. So yeah, no, like I said, I requested quotes for the tracers that David had suggested, David Drury, and I'm waiting on a response for those quotes, and we'll see how we move forward after that. I think it's just going to be faster to have them made that way than we have a lot of SAR to do here too. So yeah. Okay, great. So again, I think it was a good meeting, a lot of good discussion, and I liked the fact that we actually had time at this meeting for discussion. I think we'll try to work that into future meetings, make sure that we have plenty of time to discuss as a team. I think it's always better to make sure that we're all aligned. With that, I guess we will schedule this. Sue, you can look into scheduling this meeting, and we'll hold that as soon as possible. And we've scheduled the meetings now out until the end of the year, I think out till December, so everyone should have received invites for those meetings. Hopefully everyone can make it. And if not, then, you know, there might be potential to rearrange it, one or two of them. So let me know if you do have a major conflict. And then I look forward to next month's meeting in September after the summer vacations.
Okay, thanks a lot, Jeff.
Okay, thanks everyone.
Bye.
Okay.
Thanks. Bye now.
Thank you.
Okay. Bye.
Bye now.
Okay.